Okay, so hello everybody. I'm just going to do a short presentation to talk about genetic diversity, what we mean by genetic diversity within a population, and then just a little bit of talk about standard deviation. A lot of questions do talk about standard deviation, and this green booklet, the taxonomy booklet that we are currently on, has got quite a bit in it as well. So I just thought I'd go over standard deviation and what it means. Okay, so the picture you got in front of you is just there to show you that we are all members of the same species, but we look very, very different to each other. And to reiterate, it's not about the fact that we all have different genes, because that's not true. We all have exactly the same genes. All of our genes are 100% the same because we are members of the same species. But what's different is the versions of the genes we have, the alleles that are different. And every population holds a different set of alleles. Now, let me just clear up population and species for you. So we are all members of the same human species, but the population refers to a geographical area in which members of the same species are. So I think I explained to you, especially at the moment in this coronavirus lockdown, it's very unlikely that you are going to be mixing with people from a different country. So therefore, they are a different population of the same species. We in the UK are a different population of the same species. And it's the number of different alleles that we possess that gives us a high degree of genetic diversity. Okay, so the next slide is just saying the same thing. So the greater number of different alleles that a species possesses, the greater the genetic diversity of that species. Now, why that's important, it means that as a species, we're more likely to adapt to environmental changes that happen around us. If we have less different alleles or a reduced variety of alleles, if our environment changes, it means that there is a possibility that we might not have the alleles to get us through the environmental change. All right. Next bit, causes of variation. Now this has been thinned down on the specification now. The causes of variation we already know about because we've done a little bit about this. So the main ones you know about are genetic. So mutation will cause variation in a population. And also the things that happen in meiosis that we also know about. So crossing over and independent segregation cause changes, cause variation within a population. The reason why we don't all look exactly the same to each other. Now, in terms of the genetic changes, we can, although this is not really off spec, if I give you all the things that we are different from each other on, things like eye colour, height, whether we can roll our tongue or not, shoe size, attached, unattached earlobes, hitchhiker's thumb. By the way, if you can hear something licking in the background, that is my dog having her water, Maggie. And if you can hear the laughing, that's my daughter Grace, who's currently doing equivalent fractions and laughing at the dog. Are you okay, Maggie? Maggie? You rough? No. Anyway, we'll carry on. Right. So if you look at the list on here, I think we can separate all these things into two different areas. So something like eye colour, I don't know if I'm trying to use my pen on my screen, I don't know if you can see it. I might have to use my cursor. Something like eye colour, something like tongue roller, whether your earlobes are attached or unattached. Do you have a hitchhiker's thumb? Middle digit hair. So what that is, if you look at your longest finger on your hand, and if you look at your two knuckles, it's the bit in between that. If you turn it up to the light, you'll see a tiny, tiny, tiny little hair. And whether you're left or right-handedness. So they are all in the same bracket. If you don't know what tongue rolling is, etc., I'll just show you tongue rolling. This is hitchhiker's thumb. This is attached or unattached earlobe so far. So what I'm trying to say is, these things like eye colour, whether we can roll our tongue, whether we've got an attached or unattached earlobe, hitchhiker's thumb, middle digit hair, handiness, this is controlled by one gene generally. And so what that means is you either have that variation or you don't. So if you were going to show that graphically, it would be on like a bar chart like this. So blood group is another example of variation that's controlled by one or two genes. We are blood group A, B, or O. Okay, so things that are controlled by one or two genes, the environment can play very, very little part in changing it. Okay, it doesn't matter what you eat. If you're blood group A, you're not going to change the fact that you're blood group A. It doesn't matter what you eat. If you've got blue eye colour, you're not going to change that. Okay, it's controlled by one or two genes, and the environment plays very little part. Going back to our list, height and shoe size. Now these are controlled by many genes and our environment plays a big part in it. So for instance, if we are living in a famine area, we may not grow to the height that our genetics would dictate. So therefore, these sorts of things are controlled by many, many genes, and the environment plays a big part. So if we're going to look at that graphically, I know this is a bit blurry, we don't see that bar chart that you saw before where it's either you have it or you don't have it. We see a variation, we see a range of data between two extremes. So in terms of height, people are not 5 foot, 6 foot and nothing in between. We are a range of heights in between that. Okay, next bit then. How do we measure variation? So we measure variation by a standard deviation. It measures the spread of data about the mean. Okay, if you look at this one here, the reason why we use standard deviation, and it's really useful, if you look at these two graphs, you can see that the blue and the red both have the same mean, and they both have the same extremes, as in their range is the same. However, if you can see for the blue one, the data is got a much larger spread around the mean, and what I mean by that is we've got a lot more here and here, whereas our red one has exactly the same range, it has exactly the same mean, but if you see all the data is clustered around this mean area, so this has a smaller standard deviation. So what information does standard deviation give us? It tells us, this is for your biology questions, it tells us how reliable our means are, okay, or how different our means are to each other. This is all the questions you're going to get really on this. So if you look at this one, for instance, this is one way in which they can show it. Right, you've got two sets of data, group A and group B, as you can see, and this is the mean. Now, if you didn't have that standard deviation, you'd look at those two and you go, okay, those means are different. There is a difference between what's happening in group B compared to group A. However, what we need to look at is the standard deviation. So this plus or minus 2.7, we can add it to that mean and take it away from that mean, and we do the same here. And then we can see whether these two are sharing data. So if we added 2.7 to 21.4, that would be 24.4. Now if we took that 4.9 of 28.8, that would be 23.9. And so therefore, these two bits of data overlap each other. So what we say at this point is that the two means are not significantly different from each other because they share, the standard deviations overlap, they share data. It can be shown graphically as well, like this. So as you can see, the dot in the middle is the mean, and then this bit here is your standard deviation. Okay? I'm just pausing because my daughter is about to come in. Evie, say hi and then be quiet because I'm currently recording. Say hi. No, you can't see. It's an audio one. Just say hi.
Hi.
Right now, shut up because I'm recording. What? Sorry?
You can continue.
No, can you? No, I'm just going to pause it for a minute. Sorry, I will carry on after being interrupted. So this is another way in which we can show standard deviation. So the dot is showing you the mean, and the spread, this is our standard deviation here. Now, it doesn't really matter what this is about, but if you look, can you see, yes, these two means look different from each other, the black dot and the white dot. So the mean depth during the night, the mean depth during the day, they are different. However, they are not significantly different from each other because look at all that data they are sharing with each other. Okay, so what we say, we don't say that the results are not significant. We say that the means are not significantly different. There's less sharing going on this one, but there still is sharing. There's a bit less sharing on this one compared to the first one, but there's still sharing. And then finally, if you look at this one, S is the only one where we see a significant difference between those means because there is no overlapping of that data. All right. Let's see what else I need to tell you. Standard deviation can also look at, it's the same thing. As you can see, this is sort of correlated mean height of plants and their standard deviation, but it's showing you at different heights above sea level. And so therefore what we can see is there's a lot of variation in the plant heights when it's not very high above sea level, when the altitude isn't very high. But as soon as we go to quite high levels, the mean height of the plants has decreased, but so has the standard deviation. And what that shows you is when plants live in an environment that is quite harsh, they all tend to adopt the same characteristics in order to avoid to be adapted to a situation. Can you open that back door, please, and let the dog out? So if you look at site D, for instance, it's at high altitude. There'll be lots of wind, it'll be really thin soil, so therefore most of the plants adopt a very small height to overcome this. They can't grow as high and they'll be blown over if they do, and therefore there's not much variation. You know, it would be, if you imagine that on a curve, it'd be a very tight, sort of narrow curve. Whereas when they're at low altitude, there's a much bigger spread. Okay, I hope that makes a bit more sense of standard deviation and how it works. I do also remember that I didn't talk about types of, I did talk about types of speciation, but I didn't talk about types of selection when we did natural selection. So this thing called stabilizing selection and something called directional selection. If you have a look in the notes, it's quite straightforward. I think you should be okay with that one. Okay, thank you very much. There's my video. Only a minor amount of interruptions from the dog and the children. Say hello, Evie.
Hello.
Say hello, Gracie.
Hello.
And I'll speak to you soon. Bye.